Discovering a surprising twist in how our brains communicate, Johns Hopkins Medicine researchers have unveiled a previously unknown function of a protein that plays a crucial role in the timing and location of brain cell communication. This breakthrough, made through experiments with genetically engineered mice, sheds new light on the sophisticated mechanisms underlying memory formation and learning in mammals, and holds promise for developing novel treatments for cognitive disorders such as Down syndrome, Alzheimer's, and Huntington's disease. What makes this discovery particularly fascinating is the role of a protein called intersectin. The research team found that intersectin doesn't just assist cellular processes, it actively manages the position of tiny, message-carrying packets inside brain cells, known as synaptic vesicles, keeping them in specific areas until it's time to release their signals to neighboring neurons. Imagine intersectin as a kind of boundary maker, separating groups of these vesicles within the synapse, the critical communication junction between brain cells. Much like how oil and water naturally separate into distinct layers. This physical barrier ensures that communication happens precisely where and when it's needed, a subtlety that is essential for effective information processing. In mammalian brains, hundreds of synaptic vesicles cluster at each synapse, yet only a select few are actually used to send messages at any given moment. How the brain decides which vesicles are ready to fire has long puzzled neuroscientists. As Shigeki Watanabe, lead researcher and associate professor of cell biology at Johns Hopkins, explains, these tiny bubbles have their own defined domain where they prefer to stay. By holding them in specific synaptic locations, the brain can finely tune when and how it uses them during thinking and processing information. To dive deeper into this process, Watanabe and his team initially focused on endocytosis, a well-known cellular recycling mechanism by which nerve cells reuse synaptic vesicles after communication. Since intersectin was already known to be involved broadly in endocytosis and neuronal signaling, the scientists engineered mice that lacked the gene responsible for producing intersectin. Surprisingly, removing this protein did not stop the vesicle recycling process as expected, prompting the researchers to shift their gaze directly onto the vesicles themselves. Employing advanced high-resolution fluorescence microscopy, the team visualized intersectin positioned precisely between the vesicles that are active in communication and those that are not, seemingly acting as a physical divider. To further clarify intersectin's role, electron microscopy allowed observation of vesicles on the unimaginably tiny scale of 1/1000th of a meter. In mice missing intersectin, the vital synaptic vesicles closest to the cell membrane, responsible for releasing neurotransmitters, were noticeably absent from their release zones, suggesting intersectin's role is more about regulating the release of signals than recycling vesicles. To capture vesicle activity in real time, the researchers used a cutting-edge technique called zap and freeze microscopy. This approach stimulated neurons and instantly froze them at specific milliseconds to observe vesicle movements in astonishing nanometer detail. In typical mice, vesicles fused with the neuron membrane almost immediately after stimulation and were replenished within roughly 15 milliseconds, ensuring rapid and sustained communication. However, in genetically altered mice lacking either intersectin or a partnering protein called endophyllin, which binds to intersectin, this replenishment process faltered. The vesicles couldn't quickly refill the emptied release sites, severely slowing the brain's ability to maintain communication. Even more intriguingly, mutations that disrupted the interaction between intersectin and endophyllin similarly hampered vesicle recruitment, highlighting the critical partnership these proteins play in cognitive function. For the brain to process information efficiently, synaptic vesicles must be ready to act within a matter of milliseconds. Watanabe, Noted. This discovery not only rewrites our understanding of how precisely brain cells control their messaging, but also raises compelling questions. Could targeting intersectin and its partners lead to breakthroughs in cognitive health? And if intersectin acts as a gatekeeper for synaptic vesicles, what are the broader implications for neurodegenerative diseases where communication problems are a hallmark? This is the part where the science community will surely engage in lively debate and far-reaching exploration. Interested readers are encouraged to share their views. Do you think proteins like intersectin might hold the key to future brain therapies, or is this just one piece of a much larger puzzle? Your thoughts and perspectives in the discussion forum below could shape the next wave of neuroscience inquiry. Reference Ogunmowo, Th. et al. 2024. Intersectin and endophyllin condensates prime synaptic vesicles for release site replenishment. Nature Neuroscience.